and we're now heading to the final talk for the session. This talk will be given by M.C.W van Rossum from University of Nottingham, and the title of the talk is Competitive Plasticity to Reduce the Energetic Cost of Learning.
Okay, it's great to be here. It's a fantastic meeting so far. So, yeah, we've heard a lot about energy, and of course the whole thing is that the brain uses energy, but it doesn't have so much energy, so it needs to be very efficient in processing, and realizing that has not only informed solutions that we have seen at this meeting, how to do more efficient processing, it has also given us insight how the brain actually organizes things, so it uses sparse coding, for instance. All fine and good, but maybe we're missing an important part of this equation, and that's namely the energy you need to learn something. And that energy can be quite substantial. Here's an experiment that was done on fruit flies. The fruit flies are exposed to an odor of a flower, and then they get a shock. You do it a couple of times, and these fruit flies might be small, but they're not stupid. And next time when they encounter the odor, they will go the other way. So they learn this. All fine. What they then did is, in this experiment, they stopped feeding the fruit flies. They stopped feeding the fruit flies, and yes, they die. And so you measure simply how long it takes for the fruit flies to die. In the white bar there, that's the flies that were exposed to this protocol. They had learned the association. The red is, for instance, a control case where you don't pair them, so they don't learn the association. And you see the difference is about 4 hours on a total lifetime of about 24 hours. So that's indicative that there might be a lot of energy associated to learning. Here's another thing you can do. You can just be a bit nicer to the fruit flies and give them as much sugar water as they want afterwards. And what you see is that after learning, they double their sugar intake. And if you have school-going children and they're going to a decent school, you might recognize this phenomenon, right? Children coming home from school are hopefully hungry. Another variant of this experiment is to first make the flies a bit hungry already before you try to teach them this association. When you do that, the fruit flies, they say, screw it, I'm not going to learn it, because if I'm going to learn it, I'm going to die of starvation, and maybe I'm going to be happier dying of the stimulus than of starvation. So there's actually an interesting neuroeconomic dilemma there. What should it do? Should it learn and avoid the stimulus, this hazardous stimulus, this shock, or should it choose to not learn? So there's an interesting dilemma there. We have a poster on that where we work this out in a reinforcement learning framework. Right, so how much energy is there actually? You can calculate, make some simple estimates, and we calculated that the fruit fly spends about 10 millijoules per bit of information storage. In this simple association protocol, there's about 1 bit of information. That's quite a bit worse than your SSD drive or your DRAM, which are on the order of picojoules per bit. But that said, although these electronic costs are way more better than the biology, I wouldn't be surprised, and this is a prediction, that before I retire, so that's my trick to do it. Before I retire, the energy costs for learning, if we move away from von Neumann architectures, these energy costs for learning are going to become important in machine learning as well. By the way, the memristors, as I learned at this meeting, and maybe I make a mistake here, so please attack me and come and see me, but I think they're somewhere in the order of 100 picojoules per bit, or so somewhere in the middle of this. Right, so we want to do the following. So, MNIST, we've seen that before, right? So, if it's already so expensive to learn such a simple association, how much energy does it cost to learn something more complicated? So, something like MNIST. We use a hidden layer setup with a variable number of hidden units. We have a mean squared error function because cross entropy leads to large weights, which is inefficient from an energy point of view. And we use typical backprop, and we don't use batching, but I have something to say about that at the end of the talk as well. Before we then can start to know how much energy training such a network costs, we need an energy model. And of course we go to our biology friends and we ask them, why is learning so expensive? And unfortunately, it's not known at the moment. It's one of the big unknowns in this field. It's not clear why learning is so expensive. There's many candidates. It could be protein transport, it could be receptor insertion, it could be replay. We're going to assume in this model that it's the synaptic plasticity that's costly and there is no cooperative or competitive effect between the synapses. To say it in a physicist way, it's extensive in the number of synapses and the number of updates, or to say it slightly more understandable, the more updates you have and the more synapses you have, the more expensive it's going to be, and it's going to be linear more or less in that. Then we still have to make an assumption how we're going to then count what one single synaptic update costs, and we take two examples. We take what we call the M1 energy, and we just take the amount of synaptic update, whether it goes up or down. We don't care about that. And the bigger the update, the bigger the cost. And it's as if on that cartoon on the right-hand side, we just count the total length of all the red bars there. We also take the M0, which we just count the number of updates, just so the number of red line elements there. And of course, for the mathematicians among you, this is just the L1 and the L0 norm of the gradient that we calculate. Right, so if I give you a task to build an MNIST network, you of course make a network with maybe 100, 200, maybe 1,000 neurons in the hidden layer. Biology doesn't have that freedom, right? Our brains are given a certain size, and now backprop tells you that you should update all your synapses for all your samples. And that means large networks are very expensive to train with traditional backprop according to these energy measures. You see here some simulations, and then I've extrapolated it to the number of synapses you would find in a macaque V1, and these are really very large numbers. It more or less increases linearly for this M0 energy. It more or less increases like the square root for the M1 energy, because the updates get smaller if you have bigger networks. So that's bad news. We want to get this independent of the network size. It would be very annoying if biology would pay a price by having bigger networks. In other words, if we have here our friend Mark Zuckerberg, he already bought one nuclear power plant. Maybe that's enough for him, right? I think the world would be a better place if there would be just one power plant for Mark Zuckerberg. So, how are we going to do that? Very simple. All the stuff I'm going to talk about is very straightforward approaches. Can we just mask the plasticity? Yes, we can mask the plasticity. And you can just, before you start your simulation, you can say, well, these synapses are going to be plastic and these ones are going to be static. And you have to kind of course, then find the right ratio of that. And you can then train your network that almost flattens out these curves. So now the learning becomes independent of the network size. But you see for this M1 energy, the bottom plot, it still goes up a little bit. We didn't like that, so we wanted to get rid of that increase there as well. That's also possible. You start to coordinate the plasticity, that you say the output synapse is only plastic if the input synapse is plastic as well. And that way you kind of create paths through your networks, plastic paths through your networks, and then indeed the energy becomes independent of the network size. You can do it even a bit better. You can make it competitive. So rather than at the beginning of your simulation say, these are plastic synapses, these are static synapses, every trial I'm going to look at where the gradients are biggest and where they're smallest, and the rest I zero out and I only update the ones with the largest gradients. That works also well. And again, if you combine it with this coordination trick where you say input plasticity means output plasticity, then you get the lowest energy possible. All right, there's also another advantage for this competitive plasticity compared to these fixed masks. If you keep on moving around where your plasticity is, you can have actually fairly small network with only a very small fraction of synapses that are updated all the time. And if you wait long enough, it will still learn your task. So you can, if you're power limited rather than energy limited, you can still learn tasks on small networks. I told you already that backprop tells you to update all the synapses on all the trials. And so here's traditional backprop. If you say that thing is a 7 there, backprop tells you, yeah, well, are you 100% accurate? And you say, no, only 90. Well, then backprop will give you an error signal, right? Will give you a gradient, then you have to update your synapses. Can't we simply skip over those updates? Because we're already very good with them, right? And this was an idea that Aaron had, and we have a poster on that as well. And it's actually possible, but you have to do one little trick. It turns out, if you only train when you make a mistake, you become quite fragile in your performance. But if you remember all the things you did wrong in the past, so a nightmare scenario maybe, you remember everything you did wrong in the past, which is going to be a small set of your total MNIST data set, then you actually can save substantial amounts of energy. This is okay for the regular MNIST data set, but it gets better the bigger the data set is. And so there's on the extreme there's the extended MNIST data set, which is just MNIST with 4 times as much data points. And we find there an order of magnitude in saving. This, by the way, is really not only algorithmic kind of a theoretical saving. This is really the time it takes on our computer to run it. This is without batching again, but we have some ideas how to extend it to batching as well. Right, so those are 2 simple ways, sparsification in space, sparsification across training, that help you to save the number of synaptic updates and thereby make learning frugal without really impacting performance. There's also biological evidence for these kind of sparsifications. For instance, if you try to potentiate a synapse twice rapidly in succession, it will not do it. You will give the synapse some rest for about an hour before you can potentiate it again. In particular, for these long-lasting forms of plasticity, this is the case. Also, when you repeatedly do pairing, for instance, of pre and post, you might expect from just interpolating in your learning rules that the synaptic plasticity would slowly creep up. That's not true. It keeps on the same level for a long time, and then suddenly it makes a quite large jump and then stays at the next level. And this was first described actually by this nice paper by Peterson. It's later been seen also by people like Wang. Right, so some biological evidence for this sparsity of plasticity. I showed you this slide already. It shows you that if we look at electronic systems, the more persistent forms of plasticity, SSDs, are more costly than the less persistent ones, the transient forms of memory. And this is something that also holds in biology. Also in biology, we have multiple forms of plasticity. We have so-called early phase LTP and late phase LTP. The early phase LTP is typically protein synthesis dependent and lasts forever, whereas the early phase LTP decays within an hour. And these have big differences in their energy requirements. This is the same plot that I already showed you the left side of. On the right-hand side, you see now what happens if you do this early phase LTP, or its equivalent in flies. And there you see if you do these masked protocols, which only lead to early memory expression, there's almost no energetic cost. So you can use this. And we propose an algorithm that's been published now a number of years ago, which we call synaptic caching, where you basically accumulate all your updates, and then once in a while you consolidate those updates in long-term forms of memory. And again, you see you can get easily an order of magnitude in saving. All right, I think that brings me to the end of my talk. I think there's convincing evidence, definitely from the fruit fly literature, that learning is metabolically expensive. Of course, people have also looked at this in mammals and humans. There's also evidence there. It's less clear, but I think there's good reasons why it's less clear, and we can discuss them in detail. Understanding that there's a pressure on plasticity cost in biology, I think will hopefully help us to better understand why plasticity in biology is as it is. And hopefully we can also use this insight to build better hardware, whether it's neuromorphic or not. And I showed you with the example of skipping over the already correct samples that this is indeed possible. With that, I would like to thank you.
Thank you very much for this very interesting talk. Do we have questions from the audience? Maybe while questioners are lining up, one from my side. This final idea that you presented, the synaptic caching, to me it sounds a little bit like mini-batch gradient descent.
It is very similar. It is very similar. You can use it almost like batching. Yeah, and we actually, in our paper, our original version, we had a batching-like figure. And it is like a batching system. It's slightly different in the sense that in batching, you wait till you express, really, right? You first accumulate and only then express. Here we have a continuous buffer, so it's a bit more almost like the fast wait stuff by Jeff Hinton, and yeah, it's definitely comparable to that. Yeah, so you want to kind of accumulate, because if you don't do that, what happens to the weights? They really go up and down all the time, and that's very inefficient, right? So you want to average that out, mini batch, averaging, once in a while consolidate. Yes. It would be also interesting, of course it requires more complexity on the synapse, right? And it would be interesting to think of memoristic devices, say, which have both components, which have both an early component which is cheap and decays maybe a capacitive type of memory, and then combine that with a more persistent form.
Very, very interesting. Thank you. Okay, let's start here.
Okay. Hi, my name is Dennis Lai. I'm from TU Delft. And I was wondering if we combine this idea of like energy efficiency with this predictive coding talk we heard earlier. I was wondering, for example, if you have, like in the visual cortex, you have different layers, basically of a hierarchy working together. If you could, for example, if every neuron has its own variance and some part of the brain is aware of every neuron's output, then this could keep track in a way of all the variances, and maybe when you make a wrong prediction, only the neurons with high variance could receive an update or be forced to adjust.
Yeah, this might work, yeah. It's not something we've tried, but I think the general, I mean, I've showed you basically one application. I think many learning algorithms would be interesting to see if you put in this extra constraint, how can you use these type of saving strategies, or do you need to invent something else? Yeah.
Like a small addition, maybe, for example, then this teacher, maybe, that updates could also be aware of the context. Like, for example, we are hungry, we are low on energy, so let's not update it, or just a little, or something like this.
Yeah, yeah. And of course, there's reward signals in the brain, and all these late forms of plasticity that I showed you, the biological forms of plasticity, they're typically dopamine gated. So without dopamine gating, or probably maybe some other messengers as well, you typically don't get long-lasting plasticity. So and these feedback signals that you talk about, yes, in the fly, they know the precise neuron that tells the fly whether it's hungry or not, and whether to express this fly with all the genetics, of course, whether it's going to express long-lasting memory or not. Yeah.
Okay, thank you.
Thank you. Then let's have the next question.
Yeah, thanks for showing us how we can nicely save energy here, but what impact do your methods have on the training accuracy, on the maximum performance you can reach? Especially maybe on something more complex than MNIST.
We haven't done many more complicated tasks. It's really a question of trade-offs that you have to make. So the first method that I showed you is when you have your network is very large compared to the task complexity. In that case, you can save a lot of energy. If you have a network that is very small, then there's not much to be saved there. And the other one is about the kind of data set complexity compared to the task complexity, right? If you have a very overcomplete data set, you can make a lot of savings. And so these give us indications whether or not you can make savings on the, whether you impact the training accuracy with these methods, right? Whether there's anything to be saved in the first place, right? You can always set the parameters so that you say, okay, I don't do any savings, but then and you know, make it very small. But it's really this question of this trade-off between these quantities.
Thanks.
Thanks. And a final question?
Yeah, thank you very much for interesting presentation. I understood how you measured the cost of learning in biology, but I didn't understood how you convert it to bits. So this 10 millijoule per bit is
Well, this is an estimate, you know. These are really very much ballpark figures. And the idea is that when you do a associative conditioning, that's basically the fly can do two things when it sees when it smells an odor. It can approach or avoid. It has just two pathways. These are well mapped. And so it's just a one-bit information, basically, that you learn. Like, this particular odor, should I attach the approach or the avoid bit to it? And that's where the bit comes from. Maybe in practice it learns a bit more, maybe a few bits, you know, but it probably doesn't have a very complicated memory like, oh, I remember I was in this particular experimental chamber and this experimentalist came along and gave me this odor, and then I was shocked, and I'm going to remember that as an episodic memory, you know. It's probably not like that in the fly. So we can, I mean, these are ballpark figures, but that's how we come up with them, yes.
Thanks.
Okay, so thank you very much again, and let's thank all the speakers of the session.